So, hello all. In this lecture, I will be briefly discussing about speckle imaging technique, which is the last topic on the speckle-based measurement techniques, which we were discussing for a few lectures. Um, this is an imaging method where we don't use the principle of interferometry. It is purely based on the intensity measurements recorded with the speckle image. So, as I mentioned previously, that speckle used to be considered as the noise source in an image, where it is a random distribution of intensity uh, governed by the spatial statistics, and initially it was not understood clearly what is its origin, and later on when a lot of studies went on, it was understood that it is basically a signature of the object in a particular state. So if you can locate the variation in the speckle image, one can easily locate the variation in the state of the object. Based on that, we we discussed the principle of speckle interferometry and shearography in the previous lectures. So in this, we are going to discuss the imaging method where there is no interferometry involved. As you can see in the experimental setup, you have a laser diode which is expanded, and the expanded light beam is illuminating the surface under investigation, and then whatever light being scattered generates a speckle image on the digital camera. Okay, now what you do is, in this case, previously we used to record the interference pattern, but there is no interference involved here. It is purely imaging based method where you record multiple images over time as shown in this figure, right? So this is the first frame, second frame, like this. Now each frame consists of the speckle image of the object at different state. Now imagine the object is not going any change while during the measurement. So all of these frames will look exactly similar, right? But if there is any change, then the speckle image will differ based on the change in the object state, right? Now based on this principle, what you can do is you can analyze the variation in the intensity at each pixel. Let me state again, we can analyze the variation in the intensity at each pixel and find out how much change the object has undergone at that location of that pixel, right? So if you can do that, the same thing for each pixel, one can easily find out the speckle contrast map. What do we mean? We obtain the speckle contrast map, which indicates the different areas having undergone different amount of variation. Okay, different amount of variation. So some regions may be such that they don't go any change, undergo any change, whereas other regions may undergo a significant amount of change. So that change in the activity on the object surface can be detected based on speckle contrast image we obtain when we analyze the speckle images over time. Okay, and that's why this method is known as laser speckle contrast imaging. It is a very mature technique, especially in bioimaging, which is used for imaging different type of uh, biological objects, and especially during surgery, it has been used as a feedback mechanism to take particular action, control action in the operating conditions. So it is a very I will show you a very nice example of laser speckle contrast imaging with a leaf. So this is available on YouTube. You can have a look at that. So you can see that a leaf is being kept on a flat surface, and it is illuminated with a laser light source. So you can see that it generates a speckle image. Okay, it generates a speckle image, and you can see that there is a variation in speckle activity along the vein. However, other regions are not indicating much variation in the speckle image. as you can see here, this region, right? So when you analyze each pixel, you can generate such a speckle contrast image, which will give you a nice idea about which region of the leaf is more biologically active in comparison to other portion, right? So this is a live imaging, you can say. One can pseudo real-time imaging of the speckle image of the leaf, which one can utilize for the analysis purpose. Okay. Now, moving further, how do we generate such images? How do we generate the speckle contrast image? Typically, the laser speckle contrast imaging basically works with the speckle contrast, which a single image speckle contrast is obtained using this formulation where it is sigma divided by the mean value of I, where sigma indicates the standard deviation. Okay, and standard deviation of the speckle image, and I represents the mean intensity. So if you take the ratio of that, then we obtain the speckle contrast. Typically, the speckle contrast lies between 0 to 1, where 0 indicates decorrelated speckle pattern and 1 indicates correlated speckle pattern or fully developed speckle pattern. Now, in order to generate better speckle contrast, to see the images which we just saw with the leaf sample, two popular methods, although there are different methods, even in our lab, we have published a paper which where we proposed a speckle contrast imaging method using singular value decomposition of the speckle images. I have not included it here because it is slightly involved in terms of mathematics. So I'm not going to discuss that, but the two other methods which are also quite popular, and we had shown that our method shows better contrast in comparison to these two. So if you see these two methods, what they are doing is, you can see, considering you have n images, so n is number of speckle images. So imagine you are recording a video. Okay, in the video file, you have n number of speckle images that are being recorded. So what is done here is that, as you can see, the first of all, first image minus second image divided by first image plus second image is done. Now this is done pixel wise, as you can see. For each pixel, this is performed, and we take the ratio of this. Okay, and you do this for all the pixels or all the frames. So I1 minus I2, then you do it for I2 minus I3, then I3 minus I4, like this for all the images, and you accumulate this ratio and generate a image which is known as Fuji method based speckle contrast image, right? And the other method is known as generalized difference method. So why it is called a generalized difference is, as you can see, this is a ratio of difference between two intensities and the summation of two intensities. In this, you take only the difference, but in this case, as you can see in the Fuji method, we take the intensity difference between two consecutive frames, right? Intensity difference. However, in this case of GD, you not only take the difference between two consecutive, but the difference with other speckle other frames as well. So you can see that if I take the first image, I let k is equal to 1. So this summation is I1 minus I2 plus I1 minus I3 plus I1 minus I4, like this, right? And once you do that, so this summation is up to I1 minus In. Okay. Now you take another summation, that is I2 minus I3. We have already done I1 minus I2. So I2 minus I3 plus I2 minus I4, and you go up to I2 minus In, right? So with this, and all of this sum are again, all of this sum is again summed together, as you can see, right? That's why this method is known as generalized difference, where difference between uh, each pair of speckle image is being considered in the calculation of speckle contrast. So with this approach, you obtain the generalized difference method based speckle contrast image. So with this, I will show you some examples here. So this is one of the speckle image uh, obtained from the brain imaging, I think, of a rat cell. This is the, so if you look at this website, this data set is available here. Okay, so in your assignment, you have to download this data set. So this consists of n equal to 16 number of speckle images. Okay, 16 number of speckle images. So when you apply Fuji method, you get this image which indicate different blood vessels, right, which are clearly seen. Even you can see the contrast between the blood vessels, right? And this is the one which is obtained with GD method, right? Now if you look at only the speckle image, you don't see anything. However, if you, and this is a random image, right? Though no information is can be deciphered from a single speckle image, but however, if you analyze this over time with 16 images, you obtain this. On the other hand, so I have only one to show here, but I will show you some MATLAB demonstration for this. So for the same thing, for the same data set, when I do you can see I have, for the purpose of demonstration, I have shown two speckle images. This is at time t is equal to 0, this is at time t equal to 1, or t equal to t1, okay, t0 and t1. So you can see that you don't make out anything. There is a some variation may be there, but you cannot make out where the difference has occurred just by visualizing. But when you analyze it using Fuji method and GD method, you get a nice speckle contrast image indicating the presence of blood vessels, right? So these blood vessels are the region where the activity was minimal and activity of speckle, whereas this region it is maximum. That's why you can see the difference between speckle contrast. Now there is another method where another object for which the similar analysis was performed, but instead of the blood vessels, here a maize seed data, there is a seed which is studied. Now depending on the quality of seed or the amount of water content or other content in the seed, the bioactivity on the surface of the seed will change, and that is being mapped using speckle contrast imaging. So in this, you can see that these are the two speckle images of a seed, right? So you can see that this is the speckle image. Now the difference in speckle contrast. Now here, as a image processing expert from the course point of view, you really don't know what is going on in this region, but the expert in the field of agriculture will be able to tell what does this speckle contrast is conveying. Okay, now GD is conveying a different information in comparison to Fuji, because you can see that this region is highlighted by Fuji, whereas this region is not as highlighted by GD as compared to Fuji method, right? and you can see that the region where there was no speckle data, you don't see any speckle contrast. It is a flat region. So such this approach has been utilized for extensively utilized for the characterization of seeds. That what is the quality of seed without cutting the seed or destructing it, you can do that using speckle contrast imaging. Note that similar to other methods, this is a non-contact method and non-destructive method. You are not disturbing the object and you are not destructing the object also. Not touching the object during the imaging process. Plus, this technique has also been used for studying the. We also did some experiment on that, studying the freshness of fruit. So, if the fruit is fresh, then there is more bioactivity on the surface of the fruit due to large amount of water content in that. But as time progresses, as the fruit gets old per se, the water content reduces and the bioactivity reduces, and that correlation is plotted. So I have not plotted the correlation here. One can also plot the correlation between two speckle images and plot a diagram of correlation versus time, and one can easily see how the speckle images get decorrelated over time based on the amount of bioactivity. So this technique is also used to study how the paint behavior changes when it is applied on a surface. So depending on the surface quality, the correlation between speckle images obtained on a painted surface also changes. So such things, like you can think of any such kind of application where you can correlate some physical quantity with variation in the speckle image, and you can utilize the same. Recently we have published a paper where you can measure the surface roughness at micron levels. Especially it is used for machining operations. You might have done some operations in your first year course, such as milling, drilling and all. So once you do these operations, you want to know how smooth the surface has become, what is the surface roughness. So that can also be calculated, that we have proposed a method using speckle image. So another example I would like to show, which we had obtained from one medical institute in US, where they did the experiment with retina to image the retina using speckle imaging technique. So this data was collected there and we used it. I'm showing you the data set, so it will take some time. Because here we are using 80 images. In fact, there are more than this, around 160 or so. But I'm using only 80 images for the purpose of demonstration. As you can see, these are the two representative speckle images out of 80, and these are the speckle contrast images. Now you can see GD is not that good, contrast is not that good. However, with Fuji, you can see different blood vessels connected to the retina, right? So such type of imaging is possible with speckle based technique, right? So with this, because I just wanted to introduce this part which we left in the previous lecture, I thought of completing it before moving on to the next topic of speckle. Now you can see that here the speckle is used as the measurement tool. On the other hand, when you go to speckle interferometry, okay, the speckle image. So let me show it. You can see here, in this context, I treat speckle as noise. Okay, in this fringe pattern, speckle is a noise for me. I wanted this region to be completely white, but it is corrupted by speckle noise. So in this case, we would like to apply some denoising technique to regain the fringe pattern without noise, right? So how to apply these denoising techniques? I will discuss few most popular denoising techniques in the next class. Thank you.